The next talk will be given by Ron Elber from University of Texas, Austin. Ron is going to talk on modeling membrane permeation. We could see your slide.
Thank you. So what I'm going to try to describe today is the research that you're doing in the last couple of years, and this is modeling membrane permeations. I will focus on one type of molecule, which is peptide. And in the cartoon here, what we see is that membranes are having a very complex structure with a lot of things, biological membrane, with a lot of things embedded in it, like channels, signal protein, and so on and so forth. So there are a lot of transfer that is done by the membranes in a controlled way by passing, for example, through a channel, but there's also what we call passive permeation, in which molecule of different size on a different complexity, just passing directly through the phospholipid and from one side to another for various purposes. Viruses, in some cases, they are indeed passing directly through the biological membrane, and that example is, for example, is the HIV virus. I will going to focus on one particular peptide, which is just sketched in here, and we'll talk why this peptide is so interesting. I want to emphasize that the fact that peptides are so efficient, that there are a class of peptides that are so efficient by passing through membrane and doing something in the cell is actually quite surprising. The cell has a lot of machinery to degrade peptide very, very quickly, and the lifetime of particular peptide when you inject them into a living system is less than 20 minutes. So somehow they have to establish a lot of things doing this type very, very quickly. And this is short peptide and of orders of 30 amino acid. That is, it doesn't have a particular structure in solution. So it sounds like nothing interesting is likely to happen, but somehow it does doing a lot of things. So in fact, there are a lot of peptides that are called CPP in short, or cell-penetrating peptide, that in many cases, they can be agents of cells death. So somehow they can cause a different type of cells to die in very different mechanism. All of them are roughly of 30 amino acid long, so they are relatively short. Some of these peptides are antimicrobial, so they are killing all kinds of bacteria by the peptide is doing something to either to the plasma membrane or inside the cell. There are some peptides that are anti-cancer, and therefore they are very interesting from medical point of view, and are called ACP. Some peptides are both. So some peptides are both antibacterial, antimicrobial, and anti-cancers, and are of particular interest. The way that people find them or try to extract them from natures is they're looking for protein that serve translocation machinery. There are many proteins that by design, they need to permeate, to pass through particular membrane. And the first peptide that was discovered of this type is TAT, which is part of the HIV virus, and essentially is the leading peptide that allowed the virus to penetrate into the cell. It is highly charged, and it's all arginine, and it took a while to figure out what the hell is going on in this particular charged peptide, given that the membrane is really made of hydrophobic environment, at least to the center. So most of the peptide, again, 1,700 of them that are known today, are derived from translocating proteins. And by given a particular CPP, we can use it also to carry certain cargo to the cell, and that is one of the ideas in which people are using CPP to try to cure cancer by peptide that aim at cancer. They're getting into the cell and then delivering something that will damage or kill the cancer cells as well. The deficiency of using CPP, and there isn't even a single CPP that is approved by the FDA as drug, is that they are, in many cases, they're non-specific. They're not really going into particular type of cell that we are interested in. And something that we already mentioned, which is the rapid clearance time. It takes only less than an hour in order to be washed out, so they have to act very quickly, or we have to protect them in some way using different mechanisms in order to make sure that they will reach their target. Sometimes you put them in another vesicle, like and target the vesicle into cancer cell and so on, but this is a whole new field out there that are trying to make them live longer and be more specific. The peptide that I'm going to discuss, that I'm going to describe some calculation on, is called NAF14467. And it is a cell-penetrating peptide that is derived with a very different idea in mind. Rather than taking a translocation machine and chopping it off, it is a transmembrane protein. It's a piece of a transmembrane protein. I have the model of this NAF1 protein that is typically found in the mitochondria. This is a model. The crystal structure is not known. But what Rachel Nechushtai, she is the experimentalist that worked on this, she chopped off a piece of this protein, a part that is transmembrane, and it's parts that is also well solvated, and use this peptide in order to do different type of experiment. And she showed, among other things, that this peptide selectively kills cancer cell. It does very little damage to normal cell, but real cancer cell, they are dying very quickly. And I'm going to show you some of a movie of a death of a cancer cell or a few cancer cells, in which we are taking a picture that were imaging that were prepared by Ron Mittler Lab in University of Missouri that collaborate with us. And essentially, you throw peptide on the cancer cell that you've seen here, and the peptide, they carry fluorescence green probe. And if you watch it, you see the green color sort of growing at the membrane of the cancer cell. So here we go. This is the movie is on a speed dial. In the actual process, it takes a few hours, but you see that the green kind of color is growing more significantly at the plasma membrane of the cancer cell. That process takes hours. It's quite surprising that it's going to the plasma membrane cell, given that the peptide is charged 5 times, so it's 5+ charge. And after a while, you see all these bursts. Essentially, the cell explode and then they die. So all these things takes hours. And the questions that, of course, we are interested to know is actually what bring those particular peptides into the plasma membrane and make them stay there for a while, and how they initiate eventually the death. So that's the task that we want to address with using atomically digital simulation. Oops, somehow. Oh, okay, sorry. So the questions that I would like to ask and partially address, okay, we have a peptide. What is the relevance of peptide sequence and secondary structure to this? As I mentioned, there's a huge number of CPP. Some of them are targeting different membranes. Some of them are targeting different cells. What are the physical forces that essentially bringing this peptide into the membrane? And can we propose some at least qualitative sequential mechanism that we can build on when we are attempting to use different peptide, design the peptide for different membrane, and so on and so forth. NAF1, for example, without any further design of what I'm just described, is passing through the plasma membrane of cancer cell without damaging the plasma membrane, and is going to mitochondria. And once it is going to the mitochondria, it is kill the mitochondria, and then the rest of the cell die in the same picture that we have seen. So the peptide is positively charged. So how 5+, how is the peptide trapped in hydrophobic membrane for such a long time, as we have seen in the movie? What essentially is the free energy barrier? Because, you know, ours is a long time, and you can argue that this is a rare event for this small peptide to go to the other side. And if we really would like to know what does it do in order to kill the cancer cell, but for that, I don't have an answer, so we leave this as an open question here. We want to simulate this peptide membrane system using a atomistic model to try to get the best insight that we possibly can. And there's a number of challenges. The obvious one, that this is really a very long time process that extends on many, many scales, starting from nanosecond, in which the peptide is attached to the membrane, and attached to the membrane surface. That's the typical time to take, until hours and even days, in which the entire process has been completed, that the membrane went into the membrane and then leave from the other system. The system is large. I mean, and people have done larger systems these days, so this is perhaps not the most important factor, but nevertheless, you can go into hundreds of thousands of atoms, and you are really trying to model well the membrane and the actual solutions and so on and so forth. It's a very complex environment. When you pass inside to the membrane, this diversity of different environments is definitely far from homogeneous. It's going to change as a function of a membrane depth, as the peptide is going in more and more inside. It is partially heterogeneous. there's a different dielectric, there's different fluidity and viscosity. And on top of all these problems, if you like, or all these challenges, the peptide is that of or length of 30 amino acid, which is unstructured in water, is very flexible. And somehow we need to make sure that we are sampling at least the important conformation of the peptide when we are doing this type of calculation. What we are using a method that we developed, almost 20 years ago, and we keep developing it, but it's the method of milestoning, and it is based on certain algorithmic steps that I'm going to outline in here. It is sort of well documented, and it can be made exact, and you can also do some hierarchies of approximation, which is also useful. So the first thing that we need to do, which is a step that we can also use input from a lot of smart people around, like Mark, that can help us in order to find what are the best cost variables. The cost variable that we choose, they are really based primarily on chemical intuition. They are not rigorously being derived, but that chemical intuition, in many cases, if you have good intuition, it works quite well, as I'll try to illustrate, and we have internal check within the calculation to make sure that the collective variable that we have done are really leading, that we have used are really leading to converged result. So once we have collective variable, and here this is an illustrations of two collective variables, call them Q1 and Q2, or what we call also cost variable, we need some rough sampling in the cost space. It doesn't have, we don't do, of course, comprehensive sampling, especially that we are working in some cases in 3 dimensions, and that's not very practical, but we can still sample roughly this conformation space. And here there are conformations, or what we call anchor. These are the blue circle in this conformation stage that we sampled previously, and they serve for us as centers of Voronoi cell. It needs to be very coarse. It doesn't really have to be in with high resolution. Typically, the distances between those anchor or those centers of Voronoi cell is about an angstrom, and or less, or a little bit less. But what we do, given those anchor in core space, first, what we do, we initiate trajectory starting from the anchor, or starting from the blue circle in here. And this is the red line that we've seen here. This is a relatively short trajectory. They're of picosecond length, in which we are running them starting from the center until they hit a milestone. And milestone is an interface between Voronoi cell, like the thin blue line in here, or the mesh that we see in here. Once the trajectory terminates at a particular milestone or at a particular interface in here, we stop and we mark this milestone as active. This is milestone that we sort of sample it when we start from the Voronoi cell. And we started from the anchor at the centers of the Voronoi cell. We do it for all the sample configuration. And once we are done, we take all the sets of active milestone, and then we sample at the active milestone with a constrained sampling procedure. So we run a molecular dynamic trajectory here, which is confined into the line in here, or to the milestone in here, and we are getting a numbers of conformation at this particular point. Once we are doing it for all the milestones that are active, all the milestones that we are identified, then we run unbiased trajectory from each of the milestones, like the green line in here. We're running until they hit for the first time another interface or another milestone, and then they are terminated. These trajectories are also typically short. They rarely exceed a nanosecond. So we have many, it can go to 100,000 or up to 1 million short trajectory, and which we can done in massively parallel way, but there are quite a few of them. What we collect from this calculation are essentially this short green trajectory. We collect what is the probability that you start with a particular milestone and you end at another interface. Given that you start from here, what is the probability that you end up here? And we also collect the time which takes the trajectory to reach another milestone. These two entities are all we need in order to calculate some interesting thermodynamic variable. We calculate it so-called Kij. This is the probability that a trajectory that starts at milestone I will hit milestone J for the first time. And in the language of trajectory is essentially the numbers of trajectories starting from I and ending at J, dividing by the total numbers of trajectory that's starting at I. This is our estimate of this Kij. We also estimate the so-called TI, which is the average lifetime of a trajectory initiated milestone. We have multiple trajectories getting out from a particular milestone. Some of them live long, some of them die very quickly because they terminate on another milestone in another position. We average over all this time, and we are getting the lifetime of a milestone, regardless of what are the milestones that are finally terminated in. Giving these two functions, Kij and Ti, we can get essentially a lot of information about the thermodynamic and the kinetics of this system. For example, the one thing that we can compute is the stationary flux that is passing through the milestone. It is the eigenvectors of this matrix K, which is called Q here, with an eigenvalue of 1. So once we got this Kij, we can determine the stationary flux that passes through the milestone, which is Q. Once we know Q, we can get the probability that the last milestone that we passed is a particular milestone. And this is a scalar product of Q, the fluxes, multiplied by the time. So essentially, the way to think about this, you have trajectory that are coming into the milestone, how long they're going to stay there, essentially determine the probability that are going to be still associated with this particular milestone. So this is essentially the probability, if you like, of being at a particular milestone. And of course, the log of it, like Mark also said that you are going to see it many times, is giving us the free energy that we see in here. We can also calculate the mean first passage time, which is the probability, Q times T, divided by the flux into the final milestone, into the product state. Another thing that we can compute, which is exact within the framework of milestoning, is the commuter function, which is given by this formula in here. It's I minus K, the same K that I wrote in here. And this is a vector EF, which is 0 everywhere except 1 at the state of product. So these three functions are the one that I'm going to discuss further: the free energy, the mean first passage time, and the commuter. So the first thing that I would like to comment about is how we extract the anchor, or how we get the pictures of the Voronoi cell. This is a non-trivial process, as I mentioned, because of the complexity of the system. One naive idea that we had, we said, okay, this peptide has 5+ charges. It must really hate being in a heterophobic environment. So we put the peptide at the centers of the membrane, constrain it there, and run equilibration time, given that it is constrained in the membrane, and then we let it go. Then we watch many trajectories, which we hope will rapidly go into one side and to the other, and then we can try to look at it, and by visually inspecting it and doing further analysis, trying to understand what are good cause variable to describe the motions of this particular peptide from one side of the membrane to another. This is just a sample of one of the trajectories that we play around, and I'm playing it in reverse. So it's coming from the water until it hit the middle position in which we started with. It turns out to be very hard. Actually, the peptide likes to be at the centers of the membrane. And at the end, we had no other choice but really biasing the simulation with still molecular dynamics to get sensible conformation that we can build on, and we can build the anchor for calculations of milestoning. So as long as we have reasonable cost variable, which we extract with the help of SMD, we can do the milestoning without any further bias. So the free cost variable, essentially they are here, that we settle on, are essentially 3 distances to the membrane center. This is the distance of so-called of the leading amino acid, phenylalanine number 1, to the center, and arginine 14, which is at the end of hydrophobic helix that we see in here. The peptide does not have structure in aqueous solution, but once it is going to the membrane, at least half of it is adopt a well-defined helical structure that we see also in here. And K24 is at the other end, at the C-terminal. So we have 3 distances, and they are sufficient for the calculation we have done. We tried many other cost variables, like the trivial one, the centers of mass of the peptide, and things like this, but these 3 sort of works for us the best. They're probably not unique, but they're working for us, at least for the calculation that I'm going to present. This is the free energy landscape, and it is color-coded, and this is shown in 3 dimensions. I'm going to sort of rotate the free energy landscape in 3 dimensions a little bit so we can appreciate a little bit more the structures of it. Blue means minimum. So the free energy landscape is such that there is a minimum at the centers of the membrane, and red means high barrier, and the barrier for the entrance of the peptide are essentially at the edges of the phospholipid heads or at the edges of the membrane in order to get in. So once it is inside the membrane, it is pretty happy and doesn't require any more help. And this is sort of consistent also with the movie that we have seen experimentally, in which the peptide was essentially practically was trapped for hours in the membrane before it went on into the cell to initiate cell death. Here is another kind of a cross sections of this three-dimensional energy landscape. This is essentially the same picture that we have seen before, but this cross section is sometimes easier to see. I also drawn the same plot, the so-called max flux pathway. We now have a network because all this Voronoi cell with the milestone in between can be described as a network with edges that are connecting between the cell or between the milestone. On the network, we can ask what is the pathways of this particular network that carries the maximum numbers of trajectory, and from this extract a one-dimensional picture that can help us in interpreting the result and understand what is going on in this particular system. So this kind of a not so smooth line, that red that we've seen here, is essentially the max flux pathway. We see that it's not really monotonic in any one of the course variable, so it's not that you can forget one and get on with a reasonable calculation. There are also numbers here that I see, like 1, 2, 3, and 4. These are images that I could use to try to qualitatively explain the mechanism that I'll show in the next slide. So essentially, we are getting the free energy profile. We are getting one suggestion for the mechanism that we can explore further. And this is shown in the sign kind of a static movie in here, or with the sequence of images with the number correspond to the same number that I've shown before on the free energy landscape. So what we have in here, this is like a step 1, is the peptide lying flat on the phospholipid of the membrane. The peptide is hydrophobic, and the strongest interaction is with this arginine 14 here, which is positively charged, and the negatively charged phospholipid. And that's essentially the transition between 1 to 2 is the significant barrier that we see. And then after the significant barrier is overcome, essentially when the hydrophobic helix permeate in, then it's getting in even deeper in state 3, and until the charge group that we see in here of arginine 14 is sliding inside into the membrane. At the interface of the aqueous solutions and the membrane, now we have the end or the C terminal, which is K24 or lysine 24. And then the peptide need to do some kind of an interesting maneuver, because it need to turn around and change its direction such that it can go in a symmetric way to the other side. So there's a whole kind of a significant change in conformation that we see in here, in which the peptide is essentially completely buried with 5 pluses inside the membrane. At the same time, it distorts significantly the membrane. The yellow ball that you see here are phospholipid that is attracted inside into the phospholipid, but eventually it reverses its direction from going down. Now it's going up, and the other part of the chain is getting out. This is a cartoon model of the same thing, just to show it perhaps more clearly. And the steps are flat on the surface, hydrophobic helix coming in, then it is dragging with it one charge. The membrane simultaneously at this step is being significantly distorted to sort of compensate, if you like, to the charge in here. There are many waters, up to 1,000 water molecules that are coming in into membrane at this particular positions, and also phospholipid are getting closer. Then the helix turns around and eventually reverses its directions and moving out from the other side. Another function that's sort of telling us roughly the same story, but in a different picture, is the committer function. We can calculate the committer function, and the committer function is the probability that we are starting from a particular point that we will make it first to the product versus the reactant at every particular point on this three-dimensional cross-section of this three-dimensional shape. So the blue here means 1. So if I start from the product, this is the final escape state of the particular peptide, the probability that obviously I make it to the product is 1. If I'm starting from the reactant very, very far, the probability is close to 0. At least it's very, very low, and this is essentially is indicated on the color bar in here. But the remarkable things about this plot that I was sort of surprised when I saw it first is that extremely broad, if you like, transition state that we have in here. The transition state is around 0.5, which is the color of a light green that you see here at the center. Essentially, more than 80% of the energy landscape is a transition state, is a completely flat energy surface, which is again is consistent with the pictures of the peptide turning around. It turns around rather freely. Also, the free energy has a minimum there, and it's relatively free to move. So extremely broad transition state and barrier at the beginning and at the end, that is a kind of the picture that we have for the peptide permeation. The extremely broad transition state or extremely broad intermediate state, again, consistent with experiment of trapping the peptide for hours in the cancer membrane. Oops, what happened now? Oh, okay. So we can look at also the mean first passage time, which is another thing that we can try to compute within the milestone theory. And the things that I want you to appreciate in here is this is a log scale of the mean first passage time. And essentially, there's a very sharp increase in the time that is required to move from the last piece of the reaction. This is moving along the one line max flux pathway. We calculated the MFPT at every particular point. If you move from the edge, which is around 80, to outside to the aqueous solution, it can take you 6 orders of magnitude in time, compared to the time in which the peptide is just moving around inside the membrane. When it is trapped inside the membrane, the time scale is only of orders of 1,000. It is doing it very rapidly, many conformations that it can explore, and we can capture those kind of minimum and flexibility of the peptide within the membrane by looking at the max flux pathway and the mean first passage time that we have in here. We also have some experimental evidence for this, and this is an experiment that was done by Drexel Chad and Lorraine Webb at the UT Chemistry lab. And what they took essentially the NAF1 and they tried to attach it into a vesicle, which is a LUV vesicle. They are made for DOPC. They are very, very stable. It's very hard to permeate in, but at least they can see clearly the first step of the permeation. So first, they took this particular peptide and they put it into 2 different solutions, one hydrophobic solution, which is hexane, and then you see sort of a blue shift that the wavelength becomes shorter. And if they are putting it compared to aqueous solution or a buffer solution, which is what the HEP has seen here. Then they repeat the same experiment in the presence of the vesicle. So after a while, first, what they're seeing a shift into red, and this is the red line in here. The orange line that we see here is essentially the same as in aqueous solution and HEPA solution. The shift to the red essentially can be interpreted as the peptides situated at the surface of the membrane have strong interaction with the charged phospholipid group, and therefore make the spectra a push into the red. The probe that they put on the NAF1, I did not mention that, is attached to the N-terminal in here. It's a Danzil group. So here it sort of fills the phospholipid head group or the charged phosphate group, and therefore it is shifting into the red. But once the N-terminal is getting into the membrane in step 2, there's a significant build-up of a shift into the blue because we are now in hydrophobic environment and there's no longer interaction with other charge group. The peptide or the charge group are on themselves. So that's what I wanted to tell. That's the short story that I wanted to tell. So now 4467, 44 and 67 are the sequence positions at the original protein. Is a novel CPP. The first time we derive it from a transmembrane helix that essentially is embedded in the mitochondria and the ER membranes. And it is a peptide that is sort of consists of essentially 2 domains, one hydrophobic N-terminal segment, a helix segment, and a C-terminal segment, which is carries a total of 5 charges. It's a mistake in here. So the proposed mechanism essentially tells us that these significant barriers at the boundaries of a phospholipid head and aqueous solutions and a broad minimum at the membrane system. And it took some effort, but this calculation took us like 2 years or maybe even a little bit longer. But I think that within the milestone calculation, we were able to do that using a script, Python script that we are making available. And we are used as an MD machine, we are using the GROMACS program. Acknowledgement, and then I'm done. Thanks first for Alfredo for doing all the calculation. Rachel is the one that actually discovered this particular peptide and did many biological experiments with it. She actually can cure now mice, cancer in mice these days, and we hopefully will be able to progress even further. Ron Mittler did a lot of biology experiments, and as the movie that I have shown you, and Lauren Bebb making more of a chemical physics type of experiment that can test better the calculation and provide us with important feedback. And thanks for the funding. And thank you.
Thank you, Ron, for finishing in exactly 30 minutes. Thank you so much. So there are a few questions on the chat box. Maybe we'll take a few of them. The first one is by Anand Srivastava. I see Anand's hands are also raised. So Anand, would you like to ask the question by yourself?
Yeah, hi, thanks. Can you hear me?
Yeah.
So very nice talk. Actually, I have two questions. None of them related to rare events, I'm sorry. So the first one is what I typed in. How do you see the, the pathway that you have suggested of kind of snorkeling and symmetrically moving on the other side would change if you have multiple peptides subjected to the bilayer? Do you see any changes happening? I think you kind of answered this when you were showing some of the experimental evidence. So that is the first question. I would like your thoughts on that. And the second question is related to the imaging, the video that you're showing of the cells with the green color showing the peptides. I noticed that these peptides were not localizing in highly tubular regions of the membrane. Do they only penetrate on a more flatter surface, or is there something known about their getting into the membrane mechanism? Like how do they really do they need the tubular part, the two ends of the cells were not getting affected, and also the cell size kept shrinking. I don't know if it was right or not before it burst. So can you please shed some light on these aspects if possible?
So yeah, I mean, that's all very good question. And unfortunately, I don't have that many answers on the biology part. This is something that we still study. It depends on the type of the cancer. You can have different type of cancer cell that they respond differently to this. The peptide does not seem to go into particular positions in the membrane. It seems to be widespread. In fact, it is passing the plasma membrane without affecting the plasma membrane. The plasma membrane seems intact, just going directly through the phospholipid. What it does kill, and for that we do not have such a nice imaging yet, is the mitochondria membrane. So it is going to the mitochondria membrane and destroy it. We have guesses, but they are really guesses, and I prefer not to guess it more at the moment at this particular point. Now, the mechanism that the evidence that we have for this is for interaction is this type of experiment in here. We do not have more evidence experimentally, but we are working on it. So that's the part of the mechanism that we think quite strongly about that the peptide is getting in the membrane first. That we see also in the biological part. And the chemical physics experiment suggests that this is the first step. The rest of the step, I don't have a proof.
Okay, thanks a lot.
Sure.
Okay, so we'll go to the next one. That's from Jagannath. I'll read the question, which is there in the chat box. How does the initial SMD that you had carried out would have biased the generation of pathway for milestoning?
So it can, of course, bias. I mean, this is a very complex system with too many degrees of freedom to start with. But what we do, we have all those configuration and anchor, and we run short trajectory from them. Some of this short trajectory, they're going and finding something new that were not discovered initially. And we add them in and we repeat the calculation. So essentially, we have like a small network that allows to grow and expand. And at some point, when we do not discover anything new, then we stop. We say that we are converged. So, but it's definitely possible that, 5 angstrom away from this pathway, there's another mechanism that we never sampled. So that's the question that I cannot address. What I can address is that the network, at least that we generated, seems to be converged, is consistent within itself.
Okay, thank you. I think we have 1 more minute, so we can take one more quick question. That's from Nishant. How many course coordinates can be considered in practice? Have you tried generating biased trajectories from milestones?
So, okay, so I think that the questions of how many coordinate is important, and in principle it can be large, but we are not going to do exhaustive sampling of that space. It must be some constraint on that space such that you can really sample only a narrow band, if you like, of the full conformation space. In that case, even in the free, it can be quite expensive, but the fact that the space was relatively narrow make it really possible for us. I would say that I would not even try something more than 5. So that's just beyond what we can do.
Okay, fine. Okay, thank you, Ron. There are a couple of other questions. If you could please have a look at the chat box and reply to those questions, that will be wonderful.
Okay, I will do
this. I think please join me in thanking Professor Ron Elber for his wonderful talk. Thank you so much. Thank you.
Thank you.